Hello, my name is Jorge Barrasa Fano and I'm a postdoctoral researcher at the KU Leuven, and I'm going to talk about my PhD thesis, which is this lovely book over here. It has 160 pages and I need to summarize them in 10 minutes. So let's get to it. So, the title of my thesis is Computational Analysis of 3D Cellular Forces with Application to In Vitro Models in Mechanobiology. But I wanted to focus on two main words. Cell forces. What are these cell forces? So it turns out that in your body you have around 30 trillion cells, and all of them are able to do two things. First of all, they can change their behavior if they feel changes in the forces around them, and second, they can exert forces themselves as well. So let me give you two examples of these two things. Back in the 60s, when astronauts started to spend time in space, their bones were losing density. They were becoming weaker. This means that cells of our bones take certain decisions when they don't feel the gravity anymore. In a way, this makes sense. If there's no need to, why should cells maintain the bones, right? Another example. Many cells in our body need to move from point A to point B. Look at this beautiful video of a cell migrating and pulling on collagen fibers. It is pushing itself forward, and collagen is present all over your body. For example, we have a lot of collagen here, in this part of the ear. Okay, so far we have learned that these cell forces exist, right? But why do we care about them? So it turns out that these cell forces are very important for different diseases, like cancer, for example. So you can have a look at this video of these cancer cells, and you can see how they really migrate away from the main tumor by exerting pulling forces on the surrounding collagen fibers. So you may be realizing now that researchers are actually very interested in understanding how these forces happen, how these cells exert forces. Because if we understand how cancer cells exert forces to invade other tissues, we can maybe try to stop those forces, right? So now the question is, how do we measure or how do we study those cellular forces? So since studying millions of cells in a tissue is very complicated, researchers typically use in vitro models. And an in vitro model is basically having some cells cultured on a small dish like this one, where we have more control on the situation. And with this platform we can do simple experiments that give us very useful information. So you may be wondering now that, okay, I have these cells in this dish, but how on earth can I look at the forces that they exert? So unfortunately, we can't really measure the forces that the cells exert directly, but we can measure something else. We can measure the consequences of those forces. We do this by means of a methodology that's called traction force microscopy. And let's explain it with a simple example first, with just a rubber band. Okay, so say that we have this rubber band here, and I have drawn some dots that you can see there, more or less equidistantly, or at least I tried. And right now I'm not applying any force. Okay, so those dots have a certain separation between each other. Now say that now I apply a certain force, and you can see how those dots are being displaced. And maybe later I can do even a higher force, and you can see how those dots are also displaced even more. Now, I have no idea of how much force I did in any of those two situations, but what I can do is to compare it with this situation where there is no force being applied. And I can see how those dots had moved with respect to this configuration. And if I know how stiff the rubber band is, there are ways where we can estimate how much force I was applying in any of those situations. Now, this is a simple example with just a rubber band, but it's exactly what we do in traction force microscopy. Let's try to understand traction force microscopy with this diagram. Cells are normally seated on top of a substrate that has embedded fluorescent beads. In this illustration, they are represented with gray dots. Cells, through focal adhesions and through the stress fibers of their cytoskeleton, are able to exert a force on the substrate, causing a deformation. Note how the fluorescent beads are displaced upon this deformation. So we can take an image of the situation, and we call this the stressed state. And then we can remove the cell, and the material, which is elastic, goes back to its original configuration. And we can take another image that we call the relaxed state. So we have two images that we can compare and track the movements of the beads through image analysis algorithms to obtain a displacement field. And similarly, as we explained before with the rubber band, with this displacement field, if we know the mechanical properties of the material, we can obtain a traction field. So this was just an illustration, but you can imagine that real images have thousands and thousands of beads in them, and we need theory of mechanics and some mathematics to do all of these complex calculations. So we need the help of computers, like that one, and complex algorithms. And that's not the only problem. In the last decades, researchers are trying to make in vitro cultures more similar to the human body. We have three-dimensional bodies, right? So we also need to have 3D in vitro models. So now we need to put our cells inside a gel and not on top. Did I mention that the algorithms are very complex? Well, in 3D they become even more complex. When I started my PhD, there were very few algorithms for 3D, and none of them were made available so that any researcher or non-technical researcher could use them easily. So in my PhD, I developed an open source toolbox called TFM Lab. The main goal was that any researcher, regardless of their technical expertise with math or programming, can easily use it. It is user-friendly, intuitive, and you don't need to know any programming whatsoever. TFM Lab basically includes all the complex algorithms that are needed to visualize the forces that cells exert in a given experiment. And again, it's open source. Let's now look inside TFM Lab. In TFM Lab, we included an algorithm that recovers forces in 3D that we developed in my thesis. The main ingredients of this method are quite simple. Basically, we need to calculate a displacement field that I labeled here with a green U, which is as similar as possible to the displacement field that we measure. And I represented that with a red U star there. This, expressed in mathematical terms, is basically solving a minimization problem between those two displacement fields, and with a certain constraint that imposes the equilibrium of forces in the hydrogel. We can solve this analytically, and after some steps it turns down to just solving this simple system of equations of the form A times X equals B. And then we tested this algorithm under very realistic conditions. For example, we used a real geometry of a cell, and then we later created a finite element mesh of that geometry. And also we simulated bead images that look very similar to the ones that we encounter in a real microscope. And after simulating thousands of different conditions, we saw that our algorithm is more accurate than others that are typically used in the field. These developments allowed us to see for the first time the forces that cells need to exert to form a new blood vessel. We now have a tool ready to investigate, for example, vascular diseases. Okay. So if I had to summarize my entire thesis in just one sentence, I would say that I have developed accurate tools that allow you to extract useful mechanical information from an in vitro culture with cells, and I have made them accessible and easy to use for the scientific community.